Hi there, and welcome to this video on GCSE Biology for the AQA Specification, focusing on hormonal coordination in humans, and in particular on control of blood glucose concentration. I'm Shumina from StudyMind, where we help you revise GCSE Biology with our helpful video tutorials, tailored to your subject, your specification, and to you. If you're new here, make sure you click the subscribe button. Whilst you are watching, please leave any comments below if you're unsure about anything, and let us know if it's your first time watching our videos so we can send you our free revision materials. We also have helpful timestamps below for each part of the video to help guide you through the specification. So let's get started. Hello, and welcome to this tutorial on the control of blood glucose. So in our previous tutorial, we had a look at homeostasis in general in the human body, focusing on the human endocrine system. And we're going to apply this to the control of blood glucose concentration here, so it might be useful for you to go back and have a look at this tutorial to remind you of what we covered. We also compared the endocrine and nervous systems, and we introduced the various endocrine glands in the body. So our key learning objectives are to have a look at glucose homeostasis, also to discuss the pancreas and its function, and finally to have a look at what happens when we lose control of glucose homeostasis, a condition that we refer to as diabetes mellitus. So, these are our specification points for today. Again, as usual, just use this as a checklist so that when you get to the end of the tutorial, you know exactly what you've covered and that it relates directly back to your specification. So carbohydrates from food are broken down into glucose, which is used for respiration. The pancreas is an endocrine organ in the abdomen that controls glucose levels. So for example, glucose levels rise after eating, so the pancreas needs to act to reduce this, and we'll see how in a few slides' time. Now remember that glucose is stored as glycogen in the body, and that's stored in the muscles and also in the liver. So let's have a look at a little handy graphical representation of glucose homeostasis. So this is the range here of blood sugar your body and brain want, and just having a quick look at the axes, we have blood sugar level on the y-axis, so the higher we go, the higher the blood sugar level, and we've got time on the x-axis. This is where we ideally want to keep our blood sugar concentration, our blood glucose. However, when we get hungry, for example, we might eat something very sugary, so we're going to get a steep rise in our blood sugar. And then this starts to drop as insulin, a hormone, which we'll talk about in a little while, is secreted from the pancreas, and it removes glucose from the blood. It allows glucose to enter cells. So glucose is being removed from the blood, it falls. Now our blood sugar has crashed and we feel tired, we feel sad. So we're going to eat a little bit more and our blood sugar spikes again. So can you see how this constant spiking and crashing is causing us to have essentially mood swings? So it's really vital that we maintain our glucose within this kind of area. Now our body works really, really hard to do this. And our key organ here is our pancreas. So the islets of Langerhans in the pancreas are a group of cells that work to produce the hormones insulin and glucagon. Now within the islet of Langerhans, we have the alpha cells and the beta cells. So alpha cells produce glucagon, beta cells produce insulin. So these are our two main hormones involved in glucose homeostasis, our glucagon and our insulin. So remember, I told you at the start that glucose is stored as glycogen in the body. Now glycogen can be stored in the liver and in muscles, which kind of makes sense because we want to store glucose in an area where we can readily utilise it. So our muscles are where we need to utilise our glucose most, I guess. So we want to store our glucose in the muscles. And the liver is also another really good place to store glucose as glycogen because the liver has a very high flow of blood through it per second. So our glycogen is how glucose is stored. So what do we do when we want to move our glucose from storage into our blood so that it can be utilised by our cells? Well, what we can do is we can break glycogen down into glucose via the hormone glucagon, which remember is secreted by the alpha cells of the pancreas. The alpha cells are the islets of Langerhans. Now, what do we do when we've got too much glucose in the blood and we want to move it into storage? Well, our pancreas, our beta cells of our pancreas, secrete insulin, which then allows for glucose to be stored as glycogen. So can you see how these two hormones are doing opposite things? Glucagon is liberating glucose and moving glucose from storage into the blood, whereas insulin, so this would be the blood here, whereas insulin is allowing glucose to be moved from the blood into areas of storage where it's stored as glycogen. So as I just said, glucagon converts glycogen into glucose, so it's important in starvation and during exercise when we need to move any of our glucose stores in our body into our blood. And insulin acts on the liver to reduce the blood's glucose levels to convert glucose to glycogen. And it does this by allowing more glucose uptake by cells from the blood, which we'll see in a few slides' time. So take this example. Sarah has just eaten a very large meal. So she's got a high glucose concentration in her blood. Now, our pancreas detects this, and it's going to secrete the hormone insulin. Insulin binds to its insulin receptor, which then opens the glucose channels. So glucose can now be taken up by the cell and out of the blood. And because glucose is being taken out of the blood into the cell, the blood glucose concentration falls, and the glucose within the cell is now transformed into glycogen, which is how it is stored. Now let's look at a contrasting example. So Richard has just done an intense gym session. There is a low glucose concentration in his blood because his muscle cells have just been using a lot of glucose in respiration. So he's hypoglycemic, he's got a low glucose concentration in his blood. Now, glucagon is released from the pancreas. Remember this hormone glucagon. And this causes the breakdown of glycogen into glucose. So glycogen into glucose, then glucose passes back into the blood, and the glucose concentration in the blood rises back. So you can see how glucagon and insulin act in opposite directions. So what happens when this goes wrong? Well, in diabetes mellitus, the body struggles to reduce the blood glucose level if it rises, and this can lead to high glucose levels in the blood, something that we call hyperglycemia. So hyper, too much glucose in the blood. So hyperglycemia can lead to many problems, such as eye problems, heart attacks, nerve damage, and blood vessel damage. So it's really important that diabetics control their blood sugar to avoid all of these consequences. So how does this occur? Well, there's two types of diabetes. In type 1, the case is that insulin is not present. So the beta cells of the pancreas are struggling to secrete insulin. So in our normal healthy person who does not have diabetes, the insulin is binding to its insulin receptor and allowing for opening of the glucose channels and therefore for glucose to move out of the blood into the cell. But in our diabetic, there's no insulin present because the pancreas is not secreting insulin, and so no insulin binds to its insulin receptor, therefore glucose channels stay closed and our glucose molecules cannot move into the cell. So therefore we get very high concentrations of glucose in the blood after eating. Now in type 2, it's not that insulin is not being produced by the pancreas, it's that our insulin receptor on our cells are actually resistant. So remember here we've got our insulin molecules binding to our insulin receptor in our normal healthy person, but in our type 2 diabetic person, our insulin is secreted by our pancreas, but it cannot bind, or our insulin receptor is resistant to the binding of insulin to it, and therefore our glucose channels are going to stay closed, which means our glucose also cannot move into our cell, therefore also leading to high glucose. So to summarise for you, In type 1 diabetes, cells cannot produce enough insulin. The beta cells in the islets of Langerhans in the pancreas are not producing enough insulin. And these beta cells are ultimately, they are destroyed. Now in type 2, the problem is that cells become insensitive to the body's insulin. The insulin receptor becomes resistant. And in this case, the beta cells are overworked. So this just summarises it for you. Now this affects people of different ages. So type 1 diabetes mainly affects children, whereas type 2 mainly affects older patients. Now type 1 is a lifelong chronic condition, whereas type 2 is heavily affected by lifestyle factors. There are no risk factors associated with type 1. It's quite hard to avoid type 1 diabetes because it's often genetic. Now in type 2, there are many reasons for type 2 diabetics to be diagnosed with this disease. Now the most common reason is due to obesity, and this is hypothesized that the increase in fat blocks the glucose receptors. Now, type 1 versus type 2 in terms of diagnosis. So type 1 is diagnosed by uncontrollably high blood glucose levels. Insulin levels are low, so glucose levels cannot be reduced after a meal. Now type 2 is diagnosed in a similar way, but we can use tests to distinguish between type 1 and type 2 diabetes, to differentiate between the two. And remember, they also present at different ages. So the very young will present with type 1, whereas older people tend to present with type 2. Now treatment-wise, type 1 is always treated with insulin injections. So blood glucose levels can fluctuate, so the dose of insulin must correspond to these changes. But the person must also control their exercise and diet. Now type 2 diabetes can actually be treated by diet control and increased exercise. In particular, they have to restrict their carbohydrate intake, because remember, that's the source of the glucose. However, if this is not controlled, then type 2 diabetics may also have to move on to insulin injection as a treatment. Now, having a look at how we can diagnose, there is something called the glucose tolerance test. In this test, the person will be given a glucose drink, and the blood glucose is monitored over several hours. Now, in diabetics, glucose levels take much longer to return back to normal. So, diabetics will become rapidly hypoglycemic, so their glucose levels drastically increase in their blood. And the key fact here is that in diabetics, the glucose levels remain above normal. But in healthy individuals, insulin will be released as soon as they start eating this meal. And therefore, the insulin is released into the blood, it binds to its insulin receptor and therefore allows for glucose uptake from the blood into cells. And their glucose level rises and then returns to normal as a consequence of this. So our key take-home message from this diagram is this. So in the diabetic, our blood glucose rises and stays above normal, whereas the healthy person secretes insulin and regulates their glucose back to normal. Now let's talk a little bit more about glucagon. So the body's mechanisms are in place to increase the blood sugar level if it gets too low. And this is done by glucagon, which is produced by the alpha cells of the islets of Langerhans to increase the blood sugar level. And remember, it does this by converting glycogen into glucose via glucagon. Now, the way I like to remember this is that when glucose is gone, we secrete glucagon to break down glycogen into glucose. So when there's very little glucose in the blood, or when glucose is gone, we secrete glucagon. That's how I remember. So when the blood sugar level decreases below the set point, when glucose is gone, glucagon is released. When the blood sugar level increases too much above the set point, insulin is released. And after a meal, more glucose is converted to glycogen. The conversion of glycogen to glucose is also reduced. So you can see how this negative feedback system is tipped to balance our glucose homeostasis. So let's look at a diagrammatic representation of this. So after exercise, our glucose level decreases. Now this is detected by the pancreas. Alpha cells of the islet of Langerhans release more glucagon because we want to break down our glycogen storage into glucose, into the blood. And therefore our cells have more access to glucose and they can respire and provide ATP for energy. Now after a meal, our glucose levels rise. This is again detected by the pancreas, but this time our beta cells of our islets of Langerhans release more insulin. So more glucose is converted to glycogen for storage. And our glucose levels drop back to normal. So can you see how homeostasis is maintained here? When our glucose levels fall, we increase our glucose in our blood, glucose levels back to normal. When our glucose levels rise, we decrease our glucose levels in our blood by converting it to glycogen, and it's stored in cells in the liver and muscles. Therefore, our blood glucose level drops back to normal. So let's look at some practice questions. So a group of patients underwent a glucose tolerance test. Describe the trends shown in the graph. So pause the video here, and then I'm going to show you an example of a poor answer and a good answer. Okay, so let's have a look at the poor answer. So glucose levels increase in diabetic and healthy patients and then falls earlier in the healthy patient. This is because in diabetics, their body cannot respond to insulin, so it remains high. So have a look at the examiner's comment here. So the problem is that this answer only identifies that glucose levels fall early in the healthy patient. It fails to recognise the difference in starting glucose concentrations, and it also starts to explain rather than describe, as stated in the question. So now let's have a look at the good answer. So there is an initial rise in glucose at the start of the test. The glucose level falls earlier for the healthy patient after 50 minutes, but much later after 100 minutes for the diabetic patient. The glucose level reaches a higher peak for the diabetic patient compared to the healthy patient, and the diabetic patient has higher initial glucose concentration. So can you see how there is a description that is given? There have been 3 descriptions for the 3-marker question, and there's evidence to support it. So this is a really good example of a good answer in an exam. So use specific values. Describe each part of the curve, so the start, the trend, and how the overall appearance is. Okay, so practice question B. Explain the mechanism which causes glucose levels to fall in the healthy patient. Again, give this video pause at this point and have a go. Okay, so an example of a poor answer is an increase in glucose means that more insulin is present. This does not happen for the diabetic patient as they have a problem producing insulin. Now the problem here is that we're not talking about specifics such as the pancreas and the liver. And also the person is talking about the unhealthy patient when the question doesn't ask about the unhealthy patient. We should only be talking about the healthy patient because that's what the question is asking. So an example of a good answer to this question, I think the wrong question is pasted in this text box. So remember that the question is, explain the mechanism which causes glucose levels to fall in the healthy patient. Sorry about that. But a good answer would be an increase in glucose leads to increased release of insulin from the beta cells of the pancreas. This leads to increased conversion of glucose to glycogen and more glycogen is stored in the liver. So this is really good because we're mentioning organ specifics such as the pancreas and the liver, and we're explaining the mechanism by which the fall in glucose concentration occurs. Okay, so let's move on to practice question C. So explain the mechanism of type 1 diabetes and how it explains the trend for the diabetic patient. So again, give this video a pause here and have a go. Okay, so let's have a look at an example of a poor answer. So the insulin receptors are less receptive to the glucose and so the levels do not fall. There is also no insulin present at all and so the body cannot respond. Okay, so this is just wrong because the first sentence relates to type 2 diabetes and the second sentence almost relates to type 1, but it's not true because it's not that there's no insulin present. It's not that there's no insulin present at all, it's that there is less of it. So This is a better answer. In type 1 diabetes, the body cannot produce sufficient insulin, and so the body is unable to take glucose from the bloodstream into the cells, hence the concentration remains high in the blood. So in this answer, you could even talk about insulin binding to the insulin receptor, therefore allowing for glucose channels to open in the cells, and therefore allowing for blood glucose levels to fall via glucose moving from the blood into cells. And last question, so participants are told not to eat for 12 hours before the test. Why might this be? So a poor answer would be to make sure his insulin producing mechanisms are working well. This is just incorrect. A good answer is to get glucose to the lowest possible starting level and to ensure that food eaten previously doesn't affect the glucose levels and hence the results. Because remember, we want to standardise and for this to be a fair test. So that's all our specification points covered for today. So well done for lasting till the end of this video, but do go back. Thanks for watching this free video from StudyMind. If you liked this video, make sure to subscribe to catch our newest videos by clicking below, and leave a comment on a topic you'd like a video on. Click here to watch more videos in our series for GCSE Biology, or visit our website studymind.co.uk for free past paper compilations by topic and specification.